Hello, mic check. Okay, it's 4:03, so it's a pleasure to welcome all of you in the new year. So happy new year to all of you. Um, and I'm sure this year will be, if not as productive as last year, it will be greater and better. So thank you for coming in big numbers because typically everybody is out on the first, literally. Um, and 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 thank you for coming back for our one of our beloved own, Professor Sudipta Maiti. And it's actually a great pleasure for me now. I'll take this opportunity to introduce you because I never got a chance. And yeah, so okay. Of course, Professor Sudipta Maiti had been a faculty for almost, from 1997 onwards to 2024. It's almost 27 years, and have been an integral part of the Department of Chemical Sciences. But what I would like to go back to is refresh everybody's memory as to his educational background. So Sudipto did his integrated master's, 5-year master's program in physics. Please remember, in physics, from IIT Kanpur. And yes, and then he went on to United States for his PhD degree. Initially, he went to University of Rochester, and in 1987 he joined the program. It was, I think, a physics program at that time, but there was a lot of optics, and of course, Rochester is, absolutely popular with legends of optics. And at that time, he was going to start a program or start his problem on solitons, it seems, in proteins. But something didn't work out there. And lucky for many of us as chemists, he moved to UPenn and he worked with one of the pioneers of ultrafast spectroscopy, Robin Hochstrasser. So from solitons in proteins, he went to electrons in proteins and did some very exciting work in ultrafast spectroscopy. Of course, Robin is no more with us. But then he went to Cornell for his postdoc in 1994. So 87 to 94, and then 94, he went to Cornell, where he worked with one of the famous people in multiphoton microscopy, Watt Webb, who is also no more, unfortunately, very recently passed away. But there is where Sudipto really did a lot of microscopy and imaging, multiphoton microscopy, and did a lot of fluorescence correlation spectroscopy also. And this is where his journey with fluctuation spectroscopy started pretty much. And he carried over here back to India. When he came back, he again had a false start, just like his PhD. He went to IIT Kanpur and he was there for 1 year, and then TIFR grabbed him here. And roughly in 98, he joined TIFR and he was there till the very late of 2024. So, but he was in the chemistry department. So, and he brought a lot of biology understanding through his work using fluctuation spectroscopy and microscopy. So he served the entire gamut of sciences. So thank you, Sudipto. Of course, Sudipto, during his time here, brought a lot of very exciting, not only people, some of them who are here. First student, Parijat, is right here sitting here. And of course, the family is here as well. So thank you all for joining. But he also brought in a culture of training people the right way nationally by opening up a very nice idea of having instruments being built in a workshop setting. This was done in 2009 in TIFR. It was seeded in TIFR. And that's where Sudipto started training not just his students, but faculty members and students of different institutes. So this was a massive program that has opened up fantastic collaborative opportunities as well as attracted people from outside to again seed new techniques within the country. So thank you for that as well. And during this time, of course, academically, and I will end here, academically, he enriched all of us. He's well known for his work with amyloids, and also developing new tools to look at them in the biological context. But also he's well known to serve this forum. Sudipta was actually an NSF Colloquium coordinator. And thank you, Sudipta, for everything. And we are ready for your talk. So,
thank you so much, Jyotishman. And a happy new year to all of you. Okay, and I didn't expect this much of a crowd on the 1st of Jan because Jyotishman warned me that please choose some other date and not the 1st of Jan. But I'm lucky, and then all my friends are here. And that's actually the biggest thing that you get after 27 years of being an academic. You make friends. You make friends that you talk to and you can exchange ideas with. And once you have that friendship, then the friendship goes on. So right now I'm coming from BITS Pilani, Hyderabad, where I have been for the last 3 months now. I'm walking to my office right behind, beautiful campus. All of you are invited to visit. And so coming back, Jyotishman, thank you for the invitation to this colloquium. And let's talk about something that has been in our minds for the past few years. Okay, so I'm going to talk about, before I start, a special shout out to 2 people very close to me. One is my first graduate student, Professor Parijat Sengupta, sitting right there, visiting from Chicago. And then Dr. Orpon De, who is the last graduate student to have left the shores, visiting from Cambridge. Orpon, you're there somewhere. They're both in audience. I have one more person graduated, but is still in India, going to Harvard in 4 days or 5. Okay, so yeah. And thanks to all my students who are here. Some are still working, officially now Koti students, but still working on problems that we are grappling with. Okay, so the stuff that I wanted to tell you about is actually AI in biophysics. So there is this huge revolution, especially in biophysics, that is dependent on, and that is brought about by AI ML programs. All of you, I think, know about it for the reason I'll come to in a minute. But AI in general, if you talk to a man on the street, and I'm connected to my undergraduate network and everything, and everybody is talking about it, it's supposed to be a solution for everything, okay? So, in fact, it's too much of a solution. So, there is a hope that it solves all the diseases, aging itself. I'm talking from a biological perspective. I'm not really a chemist, so he was rubbing it in because he knows that Jyotishman knows that if he asked me to draw the structure of porphyrin on the board, I'll not get it right. So that was I could sense that. But the point is, I'm speaking from the perspective of a biologist and a little bit of a physicist, that's supposed to be the solution for everything, all problems. Okay, and soon we don't need to work anymore, and most people are convinced that that's going to be the golden world. And but some people are worried, and rightfully worried. Of course, some people are worried because they think that the AI or the bots or whatever, the robots will soon realize that the human beings are just appendages that can be gotten rid of from the face of the earth. But also there is AI in science in a big way, and in a very positive way. But there is worry that probably there is too much of it. So I'm just giving 2 examples here. One is an article where it talks about that artificial intelligence and illusions of understanding in scientific research. We're ceding too much ground. Instead of using AI as a tool, we're ceding too much ground to AI so that it's become an oracle, a surrogate for data generation, a quant for the data analysis, and arbiter. Finally, you say, okay, is this true or that true? So it has become everything, and that's a bit dangerous. And of course, in general, AI is not a solution for everything. And my son, who is also visiting from Yale, sitting back over there, presented me a book which is written by one of his friends from undergraduate friends from IIT Kanpur, AI Snake Oil. So there are reasons to worry about AI, but I will confine myself to AI in biophysics. And this story started about a few months ago, last February now, February 2024, when John Jumper was a name that probably was not very well known outside of the area of biophysics. So John Jumper was visiting us, and we decided to engage him in a debate. And the debate was specifically about what AI can't do. Ravi, sitting over there, had animated discussions with John Jumper about things that AI falls short of, and there is no direct way of predicting that the AI will get there. And of course, John Jumper from DeepMind, Google DeepMind, and he was very clear-headed about those. So he admitted some of the things, but he also said that, look, today you can't do it. Tomorrow maybe we can. So that was the discussion. We even mentioned that John, so what he did, I'll tell you in a minute, but we did mention that someday this work will get Nobel Prize. We didn't know that within 6 months he will get the Nobel Prize, and he will be known outside of the biophysics world. So let's come to what I really want to talk about. What is it that AI has done, and what is it that I don't think it definitely can't do now, and it'll be difficult for it to do it in the future? So let's look at biology, how biology works. This is a sort of idealized picture of a cell and molecules in the cell. It's not just a cartoon drawing. It's a cartoon in a sense, but it's actually taken from EM images and with specific antibodies labeling things so that you know this is the membrane of an E. coli bacterium. You see a flagellum there. Flagellum is the thing that it rotates to move from one place to other. You see this double bilayer membrane. And then you see the other proteins, and then it doesn't have a nucleus, but you see the nucleic acid, the DNA material here in the histones, etc., whatever. So we have an idea of how this machine looks like at a very gross scale. This is done in 1999, 25 years ago. It is drawn by Goodsell. And the challenge has been to get a better and better idea of how these things look. And if you look at these, most of the things which do anything in a cell day to day are the proteins, the protein molecules. So the challenge is to understand how proteins look like. This area is called structural biophysics, and you want to know how proteins look like, because if you know how they look like, perhaps you'll understand how they function. For example, something like this rotates. We know how things rotate. The car engine, it makes the wheels rotate. But how does this molecular engine rotate? There are fantastic things which do energy harvesting, photosynthesis, ion pumping, trafficking inside the cell. Cell has railroads. Krishanu has done some work on that. Photosynthesis is something that I did some early work on, but some of the work inspired by photosynthesis, some of the best work that's now coming out, photocatalysis, coming out right from TIFR, the person who is the coordinator of the Colloquium now. You have, of course, a factory. The cell makes its own string. It's not a car that somebody else makes it and puts together. It makes its own parts. So you have enzymatic synthesis. You have separation of compounds, separation of organelles, and organization overall, so that the thing actually is a non-equilibrium thing that works by taking energy from outside. All of these things, other than a few details like storing the information, etc., and a few of the ancient work for ribosomes are done by proteins. Okay, so a protein is the most important machine for cell function, and you want to know how they work. So if you look at this protein and ask how does that flagellum work, you want to know what the structure is. That when I say it's a structure, I want to know what the atomic level structure is, how each atom is connected, and then tell how it works. So this is a fantastic machine. When I say machine, it's absolutely no less than the most complex, I don't know, jet engine or whatever you have seen. There is one molecular complex. It is the bacterial flagellum. Okay, it has a hook, it has a stator, it has a motor, and it consumes energy to rotate the flagellum. It can go one directional when the bacterium senses something. One wants to tumble, it goes the other direction. Okay, so it really is a machine, and understanding life would be about understanding how each of these machines work. But remember, it's a machine, and machine is always dynamic. So how do I know, how do we determine what the structure of a molecule like that is? So for a long time, and historically, the first technique is X-ray crystallography in Cambridge, where Arpan is now coming from, but not the university, the other place. So we have, so it started with X-ray crystallographic determination of atomic level structures of proteins. Nowadays, of course, you don't do it with normal X-ray sources. You go to a synchrotron and get it done. It's a huge, I mean, it's a synchrotron. So you go to facilities, few facilities in the world, the huge kilometer scale facilities that you want. And it's still done for proteins which are large. Okay? For smaller proteins, you have a much better device now for soluble proteins. You don't have to crystallize. You can just have them in solution, more natural environment. It's NMR. For a while, I'll just keep mentioning wherever TIFR people have made some contributions. So if you just walk down outside and go towards the backside, you'll see one of these machines here. Manoj is here who maintains that. Where is Manoj? There. Okay, so, and TIFR, at least in India, led this field for a long time. And of course, the technology has gone past us. It has become, again, it's not as expensive as a synchrotron, but it's very expensive. Here you see a 1.2 gigahertz machine. We only have a 800 megahertz one. And but anyway, we did good work with it. So that's another machine for structure determination. It takes a lot of effort, money, resources, and time. One full graduate student's PhD will be done on one protein determining the crystal. And if when you're writing your thesis, if you see casually open a journal and see that the structure has come out, then you start all over again. It really literally happened at UPenn for a friend of mine. Okay, so that's that takes that much effort. And NMR takes a little less, but it's also limited to about 60, 65 kilodaltons for soluble proteins. There are some advancements, especially with Madhu and all in India and abroad with solid-state NMR, but that's not quite there yet for atomic level structural determination. Then came, and technology kept getting developed because we need to understand biology and need to understand the structure of proteins. Then came cryo-TEM. Okay, so there is, again, a very expensive machine, but not the scale of this, but a very expensive machine. We are lucky that we are I was part of leading it from the TIFR side. But remember, I've left TIFR, but you guys still are part of this consortium. It is there in IIT Bombay functioning fully. So this is, the good thing is that it can look at single particles, and by sort of shadowgraphy of single particles, you can look at the atomic level structure, derive an atomic level structure. But anyway, the whole thing is that if you want to know the structure of a protein that's worth anything, then you need to go through very expensive machines, preparations which are very tiresome and takes a lot of time. Then came, and so we had maybe a few thousand really independent types of protein structure determined. If you went to the protein database, it would be 100,000, but many of them just variations. The question is, why do we have to put so much effort into it? Chris Anfinson, back in the 50s, showed if you take a protein, it's just one single string, at least for a reasonably small globular protein, you drop it, you unfold it. It has a structure. No, it's unlike the wheel or engine of a car, it self-assembles. It's one of the wonders of nature. So it self-assembles. You can unfold it. You drop it into a jar of water, water of the appropriate kind, and it folds itself. It takes its own shape. He showed that, got a Nobel Prize for it in 1972. Which meant that all the information needed to form the three-dimensional structure of the polypeptide is stored in the specific sequence of amino acids in that polypeptide. Okay, and so it meant three things: that if you leave the polypeptide, there is an unfolded protein. This is some kind of a free energy diagram, and you see some kind of a conformational coordinate. The protein starts from something unfolded, then goes through this rough landscape, finally produces, okay, this is an aggregate, okay, intermediates. Sorry, so this is an aggregate, this is like, but it finally folds. If you have a low concentrations, it finally folds. It says, then folded structure has a free energy minimum, it's unique, and it's kinetically accessible. The point I'm trying to make is that you have a sequence, you have a structure, both are unique. So you just need a formula. Tells you how to get from the sequence to the structure. Without that formula, you have been struggling for 70 years with very expensive machines, each person giving years of his or her time in determining one structure each. So what do we do? How do we get to the formula? Well, the only thing known and the only thing that seemed reasonable is that you put some physics in it. You know, how does the string of atoms become a folded structure by itself? Well, you let the dynamics, you know the laws of physics that it obeys. So you put it in a computer and try to make sure that you compute, you let it just fold by itself and watch it. And perhaps you'll understand enough so that you don't have to do it for every protein, but you can get some general rules. The best you could do, and this is also almost a 50-year effort, and so the best you could do about 10 years ago was to come up with this very special supercomputer that also required huge amount of resources. Only D. Shaw, the guy who also owns the company, he was a professor in Columbia before, and he built this, he and his team built this special supercomputer called Anton. And for the first time, about 10 years ago, they showed for about 10 proteins or so, they could see the folding of the protein, obeying the laws of physics, starting from extended structures. So you know two things that proved. Of course, you know, it don't prove nature right. The nature is already doing it. It proved that the laws of physics that we know work, and the approximations that you put in, called force fields, are quite right. So not only you know that this protein will go into, so where is this? Something like this. These are, of course, multiple versions of the simulation. They'll go into different middle state, intermediate states, but we'll go to a final state. You know these things as well, and their statistical distribution, as you know the final state. The final state, of course, was determined already by NMR and crystallography. But the point is you have all this information. I'll come to why that is important. But you still need it, of course, a huge amount of computing power. And you could do it only for the smallest proteins. There's ubiquitin, which is some 70, 80 residues odd. And you know, it took a long time for the most powerful computer for this type to produce the result. Then came, yes.
Intermediate states. Was there any attempt earlier to capture those in a real setup while it was trying to fold?
Yeah. So there are experimental efforts and quite successful efforts for specific proteins to capture the intermediates and try to verify. So some of these are verified. Some of the structures, that intermediate structures that they did, was verified. And you can verify it with different ways in experiments. Some of our own colleagues, and I should mention, Jayant Udgaonkar, for example, has done phenomenal work again, I'm just speaking from TIFR perspective, to show what the initial stuff is. For many of the proteins, it's called a hydrophobic collapse. And you can't tell an atomic level detail, but you can look at the conformation, the hydrodynamic radius, and and then look at the time scale, because the time scale has to be right at what this happens, and these things agree. So this is quite verified, and a lot of protein folding field in experiments, I sometimes also dabbled in it, actually looked at.
Sudipta, can you elaborate a little bit, like what exactly like input? You have a number of atoms you put together,
Okay.
and you write in three-dimensional picture, and you write in Hamiltonian and some potential.
Absolutely.
And then you do time evolution and see what's happened.
Absolutely. You already said what it is. Basically, I give the sequence, which is all the bonds, and then you have the water, and with the water and the bonds, you have energy levels. You know the Hamiltonian for bending, etc., interactions
So
with the water.
potential you are putting by yourself, that's from some experimental
So
data.
these are the force fields that have been developed. Okay. Of course, the real expert is sitting right there, Ravi. But yes, you put the force fields, and getting something like this right also tells you, oops, I didn't want to go there in advance, also tells you that the force fields are reasonably correct. There are complications. I'll come to membrane proteins where the force fields are still not great. But for these, the force fields seem to be correct. And people have also got Nobel Prizes for those. Mostly classical dynamics.
So the intermediate states are the metastable minima.
Yeah.
And the final one is the global minimum.
Yes, yes. So this is the global minimum. There are many repeats of this. So the repeats are overlapped with each other. And you see some of the parts are sort of flexible, but much of the parts are overlapped with each. Every time you simulate this, they come to the same thing. And that's actually the experimentally determined structure.
And there's several hundred microsecond lifetimes, which are, these are found by the computer.
Found by the computer.
And
but some of
also agree with the experiment.
Some of these are tested with testing. You can tell states by looking at one tryptophan fluorescence, changing, etc. So as I said, you can't look at the intermediate states atom by experimentally, but those things actually agree with this. So this is experimentally very.
There's Ramiz, and then
so on a
well, let me go there first.
On a computer, I'm sure you can always find the global minimum using brute force if, worst case. But is there a situation in reality where it gets stuck in one of the intermediate states, or because there is some topology preventing it from, or something like that? Does that happen?
The landscape is rough. And if you talk about statistics, I'm sure some small fraction of the molecules, this is a very well-folded molecule. Some fraction of the molecules do get stuck for a long time, but that will be the tail of the thing. So you can also calculate the probability, because ultimately you want to fully characterize this in all its dimensions. And then you will know what percentage gets stuck where for a file. But there is something called misfolding that I work on, but I don't think I want to get into that today. But yeah, it does happen that you get misfolded proteins occasionally. Yes. Okay, he's a biophysicist.
Hello.
Ask him.
I have a more or less a similar question, which has been already asked. But how does it know? How does the computational code know that it is not stuck in somewhere intermediate, which is almost equally similar to the final state? And this can be a question of. How do you know the final?
So the real all-atom simulation, the computer doesn't try to know, it just tries to run it. But people like Ravi knows that that's very computationally expensive. So sometimes they have a way of tilting the actual free energy landscape to sort of nudge it away from those things. But he would be the best person to talk about it. But if you do an all-atom simulation without any of these things, it will run and ultimately give you that. And these guys claim that that's what they're doing with their atom. But for bigger proteins, etc., you can't do that. You get stuck for, in computational time you can be stuck for months and whatever. Okay, so this is, as I said, about 72 residues or something. Anything more than a couple hundred would be really big, and it will take very long. You need to do some. And even for this, I don't think computers available to Ravi will be able to do this in any time. So you'll have to do some approximations, but Anton could do it. But this is 10 years ago. Yeah.
So a lot of these interactions are hydrophobic. Hydrophobic interactions are entropic in nature. How do you calculate entropy in such molecular dynamics kinds of simulation?
Right. So where does entropy come in protein folding? The entropy comes in because you have a hydrophobic residue, a residue which has, say, methyl groups, etc., next to water. And the water is doing hydrogen bonding with the rest of the water molecules outside. And it can, at one side, it doesn't have the orientational freedom. Okay, so That's how the hydrophobicity comes in. So you don't have to put in a hydrophobic interaction, quote unquote, in the MD simulation to get it. It just does it by itself, and the freedom of going there, he would be the expert to tell you, is coded in. It's not put in by hand. It's not put in by hand.
Okay, one last question.
Okay. Sure.
So, unlike AlphaFold, which considers just the primary sequence, is there
That's
any?
my next slide.
Yeah.
Okay.
So, is there anything like this sort of computation which can consider things like acidity or pH constraints in the formula to predict the protein
So how
structure?
pH or pH comes in is in a protein, you have ionizable residues whose pK, the midpoint of ionization, one side will be positively charged, one side will be negatively charged in pH scale, they're known. Okay, so that's something that will come in, and you change the pH and it will change the ionizability. If you have something, say, charge, what you call salt bridges, one is positively charged, one is negatively charged, then there'll be electrostatic interaction, etc. In pH 7.4, most of the MD simulations, I think, assume what is charged, what is not charged, but because otherwise it will be a bit of a quantum calculation, which I know people do sometimes, but it gets complicated. So I think some of these are. So Ravi, you correct me if I'm wrong. But let me just go forward. So all of this took a lot of time. That's the point I was trying to make until came John Jumper and Demis Hazzabis with AlphaFold. So there was a competition that I think all of you know about, is about predicting protein structure. So experimentally, some protein will be determined, and they will keep it under covers and ask for predictions from theorists, people who model MD simulations or bioinformatic kind of people, and it's open to everybody. And you see forget this blue thing, all the other things are submissions which are wrong. This is how wrong from the right structure is, the prediction is. And suddenly somebody comes in and gets it much, much better. And it's not just a factor of 3. You see, given how big an atom is, there's a median error in angstroms. 3 versus 1 is very different. If you have a 5 angstrom level structure, you really don't know the structure. You're just guessing it most of the time. 3 angstrom, yes, I think you are getting mostly to the atomic level structure. 1 angstrom, yes, it's determined. Okay, because ultimately the protein is not a fixed thing, right? It's also doing a little thermodynamic jiggling about it. So it was astounding, and it came in with AI/ML, and that's what we are talking to John about earlier last year, and it changed everything. Okay, it changed everything because it was also, of course, they had enough computation, but it was computationally, once you set it up, computationally very cheap. So soon enough, between Google, then the race started, etc., Meta, you had hundreds of millions of protein structures solved. You give a structure, a sequence, and it gives the structure. Remember, I told you about the formula? I wanted a formula when I was growing up as a PhD student. I give a sequence, you give me a structure. Nowadays, any kid, you go in, type AlphaFold 3, you give him the sequence, you get a structure, okay, in a minute. So it's that. So it's completely changed the scenario of known protein structures, which has huge implications. And that's why people are so excited, and they say, well, okay, I mean, this is a new revolution and something coming. True, it is a revolution. There is no doubt about it. I'm not demeaning it, because if you know the protein structure, how a protein works, you also know how to stop it from working. Okay, you design something based on that structure, a small molecule, and that's a drug. Because many of the molecules, something that makes your heart pump faster, etc., you want to quieten it down. So you put a little inhibitor for it, or you put a little activator for it. So once you know the structure, you can design a drug. And this is sort of being called a miracle for drug design. I'm sure a lot of drug design efforts will be helped by this. Okay? So this is where we are right now, the beginning of 2025. So are we in the future yet? My answer is not quite. First thing is that this AI/ML thing, as John would agree, though he does local dynamic MD simulation, we realize that there's ultimately no physics here. So the dynamics part, which Anton, at least the supercomputer, tried to get it right, is missing. You can't ask for what was the structure in 100 microsecond after synthesis in the ribosome. You can't ask that. You can just say, this is the formula, this is the one-dimensional sequence, and what is the structure? And you get a structure. So there's no dynamics, no partially folded structure. This will be my point A. Whatever time I have, I'll go through point A, B, C, or drop some of these. The second thing is even more defeating. Sorry. Yeah.
Structures that this AlphaFold is giving, how many of them are is it also getting things wrong?
Okay, so
what construction are wrong?
So to make a true test, you need to go back to that kind of test where the structure is not known. But now AlphaFold is an open program. You can apply it for any protein that you know of whose structure has been determined. And hundreds of thousands of structures, the related structures, sometimes not all independent, have been determined experimentally. And it gets it right 85 to 90% of the time. So when I say right, it's not really binary. It really should be measured in terms of that angstrom scale. But when the well-structured globular proteins, it's really good. There is no doubt about it. So people tend to believe things we haven't seen. Goes back to my first slide, that AI is becoming an oracle and arbiter. But what do you do? I mean, it's difficult to determine a structure experimentally. And this guy is giving it in 1 minute, and you test it on 10 other things, and you seem to get it right. So there we are.
We will suppose I give a sequence and a pH, which will it get it right?
A different pH than the
a different pH than the normal.
So most of the structures in biology are known at pH 7.4 because that's where we are, our insights are. So most likely it would not get it right. But there are programs now coming in where they just do these charge changes and they are asking what happens in a lower pH, because some of our inside our body, some parts are at low pH. Inside the cell, there is something called lysosome, which is at pH 5, 5.5. So those things are also coming in. The point is the structures that it learns from. Where was that structure formed? If the tract structure was formed at pH 5.5, it's a lysosomal luminal protein, then it will get it right, but not randomly.
We will wait for 15 more minutes. Let him come to the point, and with the problem has been set. So yeah.
I love the questions, but we don't have infinite time. Sorry. So, okay. So the point is, if you don't know the dynamics, you don't know how something functions. It's good to know the structure. It's like looking at a car, and you have never seen a car before. You have never seen a wheel before. You have never imagined what a round thing can do. You find out there are 4 objects below this contraption which look circular. Okay? You won't know how it works. Because if I give you another car which looks square, unless you have seen it rotating and thought about it, what the dynamics is, you won't get it right. So just knowing the structure doesn't get you very far. Much of the biology, where the proteins are actually jiggling around and actually dynamic. Okay? So the first thing is that structure is not everything. Structure doesn't tell you how the things function. It's a necessity. You need to know. If you want to understand how a car works, you need to look at every part. It's not even the full car you're looking at when you're looking at structures. It's like all the parts, maybe 10,000 parts in a car, each screw, this thing is laid out on a piece of, on a blanket or something, and you're trying to guess how the car works. It's difficult because you're not seeing the car working and the dynamics. So very simple hemoglobin. If you look at it, there are 2 structures here. One is oxyhemoglobin and one is deoxyhemoglobin. The structure is slightly different. Hemoglobin undergoes a structural transformation as oxygen binds. Only with the structural transformation can it release the oxygen. Okay, it goes between R and T states. And if you didn't have the dynamics, you won't get the oxygen out where you need it. So you don't know just from the static structure how the protein will work, how the protein will function. So the function is very difficult. It's the function that ultimately you're interested in. How does it work? So it's the function. The other thing is that, coming back to the same picture that I showed 2 minutes ago, is the intermediates. Every protein starts extended. Okay, it's an experiment that we are doing with Devashish Das right now. It comes out from the ribosome extended. It's not made folded. It then folds. Okay, so there are these intermediates that it has to go through. The intermediates themselves can be very useful as structures if you knew the structures, and you don't from AI. So let's first get to that. There's a fantastic idea that's still being worked on, is that when you are looking at useful ideas, useful from a societal perspective, that you can make drugs which attack an intermediate of folding as it comes out of the ribosome. Frequently, once the protein is well folded, it's difficult to target it. Okay, but there are many more open sort of Achilles heels out when the protein is unfolded, and it is always unfolded as it's coming out of the ribosome. Okay, so if you know the things, you can. So there is a molecular dynamic simulation tool, PPI-FIT, invented by Faccioli and co-workers. They also set up a company called Sibylla, which said, well, we'll target those. So that's one of the most important things. You can, instead of one single structure, now have 100 intermediates that I can target. We did a little bit of that, so I'll also stick in some of our work. Can you perturb the folding of an unfolded protein with an intelligently guessed molecule? And our idea, the idea that Anirban Das, my student, came up with, is that so even if I have this structure, you don't know what to fit in there to stop it from folding or change the folding or something. We came up with the idea that we'll use some part of the protein itself to change the folding. See, a part of the protein is always recognized by the rest of the protein. Protein folding is a cooperative phenomenon. Okay, so if you have a pre-folded part, if you present a very nicely pre-folded part of a protein to the protein when it is folding, it cannot differentiate between its own part and what we call the xenonucleus, and it folds with the xenonucleus, and all hell breaks loose. In this case, of course, Anirban showed that protein folding can be faster if you form the nucleus preformed and supply it. And it takes a little bit of chemical intuition how to hold that little part in the structure identical to what it has in the protein. Otherwise, it's a floppy thing. No? It's like noodles unless you properly shape it. So that seemed to work. That gave the rise to the idea that Anup came up with. He's also here, sitting over there, my current student, that you can do a protein silencing. You want to stop the function of a protein. You make, so these are the two parts that say they come together, and suddenly you have, you make a copy of this because it's there, right there. You don't have to design it. It's part of the protein. You put it, and that guy will come in, and this guy will be hanging around. It's surely this will not be sitting there idle. It is going to disturb the protein to change its function. I said, wow, okay, that could be a protein silencer. That's the design is just presented to you by nature. You don't have to go for years of drug design. You can just have it. Does it work? Well, he just showed that with Devashish's lab's help, that if it comes out of the RNA and you have this pre-folded peptide, he calls SP1 there, and you are using GFP, a fluorescent protein, look at the fluorescent readout, and the fluorescent readout changes. Of course, it outcompetes. Some of its own things outcompetes sometimes the SP1, the peptide, but sometimes it does. So you get about 40–50% reduction. So you can silence a protein by looking at its own part, giving that to it, and it's a foreign part, and then it takes it, the rest of the protein wraps around it, its own hand or whatever it is, keeps flopping about, and that silences the protein. So wonderful. Can we, instead of silencing a GFP, which is good as a demonstration, but can you silence a protein of a bacterium? Well, said you try. So there is a protein for a E. coli, and is a very critical protein called BAM-A that he tried to silence just by looking at the structure, picking up one part of it, stabilizing it, and presenting it. There is unpublished data. And recent data from our collaborator, Michael Schlerf in Dresden, who does super resolution microscopy of this, shows that our peptide goes very close to the islands where these proteins are, called outer membrane protein.
What is the data?
Okay, the data. So it's a super resolution image of one E. coli. This to this is 1 to 2 microns. Sorry, you don't have the scale there. So it needs to be super resolution, looking at the localization of our peptide, which is in red, and outer membrane protein that it is targeting, which is in the green. So they are known to form. So as I said, from here to here, it will be about 2 microns. Yeah, and the super resolution gives you about 20 nanometers or so. So it's, we could build a super resolution microscope, but it's sometimes not so easy to have all the biological constructs. So Michael is good in both. So we went to Michael and he showed it. What it showed is that our peptide is targeting the outer membrane proteins. Okay, so we are very happy about that. Does it kill the bacteria? Yes, it does. Okay, and ACM done right here, and showed that the bacterial membranes are falling apart with the peptide, and just as cell viability shows. The cell viability, there is concentration of the peptide, there is how viable the cells are. 100% means control, and you see with concentration increasing, the viability drops. And this is this is the positive control. This is the spectrinomycin, the antibiotic, and this is our peptide. Okay, so our peptide is much stronger than the general antibiotic that you have for the bacteria. Still, of course, has a lot of room for improvement, but it shows that a small idea, if you have how the knowledge of how the protein would fold, can give you novel routes for finding antibiotics. Okay, so the conclusion of part A, and only have 10 minutes left, is that folding intermediates are beyond current AI, but can be observed in MD, and part of it can be also through experiments. And this can be used to intervene in protein function, and self-peptides, we call them self or zero peptides, are a simple route to design peptides that can intervene. So we are proposing self-peptides as a novel class of antibiotics. Okay, the first part I said what AI can do is not get the intermediate states, and I proved to you or showed to you, tried to show to you that they are very important.
AI do MD?
Okay, so he's trying to marry the two, so you'll ask him. All right. So AI doesn't have the physics in it. Yeah.
We'll take quick questions.
Sure.
I mean, maybe you can answer this at the end of the talk, but I mean, the AI programs are designed for very specific things, right? Whereas the picture you have presented is that, okay, there is intermediate dynamics, there are intermediate states, and then there's a final state. Whereas the problem defined, as you said, the training data is in such a way that it only looks for the final state. So if you wanted to, you could probably design something which is specifically targeting, though I can already vaguely see challenges in doing that. But are there some obviously known things which says that, no, okay, this kind of training data is just not possible to get, right?
Very good question. Yeah. And the answer is the potential energy landscape, okay, or the free energy landscape is very flat for the intermediates, okay? And the sort of goes down a funnel and goes somewhere. From that flat, I'm talking about, let's say, 2-dimensional hand waving is n-dimensional, you have little minima. Experimentally, you get signatures of those minima. But for anybody to learn, and you need atomic coordinates, to learn that, to have a training data set, right now it's impossible. I'm not going to say in server. So I always said beyond current AI. We'll see what happens tomorrow. But at the end, I'll just say that I think the time has come to generate large amounts of data and go back to AI and see what it can do. But the point is, yes, you don't have the good training data set for things which are rare.
Yes.
So
sorry.
Before you came to AI, you mentioned a Hamiltonian solver with the earlier one, right?
The MD, yeah.
Yeah. So can that do the dynamics?
Yeah.
The dynamics in that is it accurate usually?
Yeah. So that was the Anton example I showed, Anton supercomputer. That's an MD simulation, actual Hamiltonian, every twist, how much energy for every degree, every stretch, every collision, and you put it in and you make a force field. And that, at least for 10 or so proteins, what they predicted, actually are experimentally proven. The good thing is that if you go to that paper, it's beautiful because the protein never just folds and just stays there. It has a temperature. So occasionally it will unfold and come back. So they showed also, and by single molecule experiments, it's easy to know how often the protein folds and unfolds. So that matches.
So the correspondence between that model and the real protein, is it exact or is it statistical? Like
okay, the real protein means real experiments. We can tell atomic level coordinates. But as I said, I can tell the distance between two. There's a technique called FRET. Okay, so it's roughly accurate to a few angstroms. And within that, those 2 points seem to be agreeing to each other. But there is no way to, as of now, there is no way to experimentally determine all the coordinates of an intermediate.
So in the previous work, the target protein that you chose, BAME, what does it do? How is the peptide killing the bacteria?
So BAME is actually a beta barrel. So if you take a barrel, the barrel is really a sheet, and then you fold it like this. So if a barrel is folding, a sheet is folding, the last part to fold would be the edge. So what we designed, and we need time for competing with its own folding. So we designed, so what Anup did, designed a peptide, which is this part and this part. These two edges joined together, presented it, hoping that it will take so long that our peptide can get in. And so it's a beta barrel protein. What does it do? It actually is amazing. It helps fold other membrane proteins. It actually grabs them and gives them an environment to fold. Cell has a huge problem. The protein, which is a membrane protein, the environment, the cue for folding comes from the hydrophobic environment in a membrane, but the protein is being formed in solution. The ribosome is in the solution. So that has to go in to the membrane and then fold. It's not easy because it can get aggregated, etc. So it needs chaperone kind of thing. So it's a chaperone. It's a very critical chaperone. So if BAME is attacked, and there is still no great medicine, but drug, but there is a molecule, darabactin, that people have designed for BAME that seem to be showing good results. And our thing is much more general. The darabactin problem of drug resistance is that put darabactin, it works for a few years, people overuse it, and the bacteria somehow changes the structure so that darabactin never works, doesn't work anymore. And we have extended recognition, like our U-shaped protein peptide, then it's very difficult for the bacterium to change one residue somewhere, mutate one here, and get rid of this because it's distributed sensing. So I think we have good possibilities. Okay, there are only 5 more minutes, so I'll see what I can do. Okay. Okay, the main problem of AI and structure prediction is that if you look at some of our diseases, malaria, our disease, and people have been trying to look at a drug for malaria or an antibody for malaria, vaccine for malaria for ages. If you look at the plasmodium, half of its proteins are not even structured. That's a big problem. They're called IDPs, intrinsically disordered proteins. We also have about 20%, 20 to 25% of our own proteins not structured. So they're in the flat part of the energy landscape, even for ages. So there is no real energy minimum or no funnel that takes you there. And if that is the case, just the same reason that you can't get AI to predict your intermediates, you can't get a structure prediction. There is no structure, so what are you going to predict? This is a major problem. And not only for infectious diseases such as malaria, but also a protein that I have worked on for a while, which are amyloid proteins. They're unstructured. Only when there is a certain condition and concentration, they will aggregate with each other and form something called fibrils. And they're again toxic. Okay? So malaria is well known. The other diseases that the amyloids cause are also well known, Alzheimer's, Parkinson's, etc. So how do I attack that with AI? The answer is you can't. Okay? So there are some of the biggest challenges that we have now. So we go back to experiments. Solid state NMR initially was the king of looking at it. We did our tiny contribution, Madhu and I, and our students, using solid state NMR of Madhu and marrying with our optical techniques to get the structure of the fibrils. Okay. So the fibril structures were determined. So as I said, solid state NMR was the king up to about 2015 or so. And then came cryo-EM, the third instrument I was showing initially. And that sort of stole the light from solid state NMR. Now it's all cryo-EM. So Tycho, Rob Tycho was the guru of all the structure determination. He's been doing it from his first amyloid fibril structure 2002 PNAS to his latest ones. He has moved from now solid-state NMR to cryo-EM. This is the cryo-EM structure. You see how the fibrils form. Okay, they're sort of sheets. They're called parallel beta sheets. But there are some red and blue things which only now in his structure has come in. Same protein he has been working on, giving structures from 2002 onwards for 20 years. The structures are evolving. His 2002 structure doesn't quite look like this and didn't have these things called anti-parallel parts. Okay, there's only 21. Why am I talking about that? I'll tell you why. So we have spent a decade and a half looking at how these proteins may aggregate, okay? Especially the initial parts of aggregation, because many people still believe that those are the things called oligomers, not the large fibrils, which are responsible for the disease. We found out from Suman Nag, starting from Suman Nag. By the way, this work was started, amyloid beta work was started by that lady sitting over there, Parijat, our lab. Parijat would remember, and two key 2003 papers, one on methods and one on actually demonstrations. Those things blew apart the field in the sense we started doing a lot of stuff and culminated in Suman Nag and others showing that what we are getting is an anti-parallel. So I'm not going to explain it very well, but there are parallel configuration which people find in fibrils and anti-parallel configurations that you find in the oligomers. And oligomers have to be more toxic than they have to be doing something different. So we think it's in the conformations. And this is also a solid-state NMR thing, which the first proper structure of a, not full structure, but structural coordinates of an oligomer, small thing. It's extremely hard to do, but we found some signal from spectroscopy and from solid-state NMR showed that this was anti-parallel. Okay, so there was the sort of ideas of structures, and these things you never get it in AI because there are lots of different fibrillar structures, little differences here and there, and this anti-parallel thing seemed to attack the membrane. And we started, so our previous work till 2014 or 15 was in water. Then we said, well, if they have to attack the membrane, we have to get the same structure in the membrane. So this work started with Devanjan, again inventing a new technique with plasmonics and finding things. Then Bappa came in, again with plasmonics, but using fluorescence. But let me just describe what we just got a couple of months ago when Arpan graduated. He's sitting here. So oligomer, so again, there is class of structures, okay? You have a dimer, you have a trimer, you have a tetramer, pentamer, whatever. Each pentamer doesn't have to be the same structure. The next pentamer can be a different structure, and nobody knows how varied this structure is. Nobody knows what is toxic. So we said, well, let's look at how they interact with the membrane. If they can, the theory is that they will puncture a hole in the membrane. If they have to do it right, then they have to go in a particular way into the membrane. And do they go in a particular way? Can we tell? But if each is different, each single oligomer, which could be a monomer, a dying 1 molecule, 2 molecule, 3 molecule, how do I look at it? The only way to look at it is by molecule, single molecule thing. So Arpan developed techniques by something called fluorescence quenching, how deep each part of oligomers go into the membrane. Membrane itself is 4 nanometers thick. It's a humongous effort. And finally, he after developing the techniques which is dependent on the quenching, it's very simple. There is a fluorescent reporter put in, and there is a thickness of the membrane. And if it goes in here, I have a quencher by design put in here. In another preparation, I have a quencher put in here, another preparation here. So how deep it goes in depends on when it is quenched. And I know exactly how much each part of the protein, how deep it has gone in. So I won't go into the things, but after counting little dots over his graduate lifetime, he came up with this map, which he graduated, which is you look at variants of amyloid beta, some are toxic and non-toxic, and ask the simple question, do these variants interact with the membrane differently? And can I say when one single variant, is it the monomer that's different, is the dimer that's different, trimer that's different, or what it is? And so on these colorful dots, I'll just explain two. So look at the red dots are something called amyloid beta 42, the most toxic species. And this is Q-sleep value, which means is it close to the 16 means deep in the membrane, 5 means halfway there, tryptophan means above the membrane, doesn't even go in. And if the Q-sleep value is large, that means it has gone in. This is the monomer, dimer, trimer, tetramer. So it's a complex set of data for each different species, A beta 42, 40, and A beta char, which is the least toxic. You have this plot of monomer, dimer, trimer, tetramer, of how deep they have gone in. So this is all the data that you can have for part-wise identification. It's a complex data set. But you can see that there is a big difference between the completely non-toxic green and the most toxic red. You don't get the full structure that this is the best anybody can do right now about oligomer interaction with the membrane. Where do we go from there? Well, I suggest we generate a lot of this data and come back to some AI-11 program and generate a lot of variants which are toxic, whose toxicity is known, and hopefully this set of data will make a difference, because then you'll be able to predict if I put a drug X, and this pattern changes some way, drug Y, and this pattern changes some other way, we'll hopefully be able to find out which way to develop the drug. So this is the maximum information, not very easily it's not very comprehensible by human eyes, but that's exactly what AI should be good at. So let me skip the third part. So this is by part B. And so I said can generate, conclusion is disordered proteins from toxic oligomers of many different kinds. They are not predictable yet by AI. And the future would be can generate membrane interaction patterns for different types of toxic and non-toxic oligomers. Sorry to Devanjan, who is listening through, Dev Shankar, who is listening through YouTube, that we couldn't get to his very fantastic thing, which says, when no proteins are involved, there is a world beyond that. And there's, of course, AI is not yet there because the proteins are not involved. They're a small molecule world, but I'll skip that fully. And Vidita and I had a lot of fun doing this with because this had to do with psychedelic compounds. Okay, how psychedelic compounds change membranes. Nothing to do with membrane with proteins, receptor independent signaling. But I will not go there. So yeah, okay. So sorry. Here, hey, where did I go? Oops. Hold on. Let me just quickly go to the end and go to my conclusion.
Oh, no,
this one. Yes, my comp. Yeah. And my the cursor is not working. Yeah, okay. So let me reach here. So what I wanted to say is that reach of current AI applications in biophysics falls short of much that is interesting and important. Important in from societal perspective, to be able to do something with it, and interesting from just a scientist perspective. You tell me this sequence is this structure. Come on, I don't find that interesting. You tell me how it got there, I find it interesting. Then I can ask that why this and not that. Otherwise, you don't have an answer. So it's there's not so interesting, and of course, it misses a lot of important points. So innovative experiments and physics-based theory are the only ways to generate data about these things. Okay, either you simulate and hopefully the force fields are correct, or you just go like me, experiments, and generate a lot of data, and which are correct in the sense of you have repeated this and they are correct. You don't always get inferences correct from that, but that's okay. What I'm trying to say is not negating AI even in these fields. My final conclusion is that the time has come to change our experimental perspective a little bit. Previously, if a student came up to me and said, oh, I can do this and I can do that, I can measure that. I said, wait. You need to fit a model to what you're finally looking at. You have 1 variable, 2 variables, 3 variables. Don't give me 30 variables because I'll never have a model to fit that. If today a student comes to me and says, I can measure 30 different parameters and there's some relationship with this phenomenon, I will say, go ahead. Go ahead, because with AI ML, I think we need those. Right now, is a partnership between UK and DAE. I'm building a microscope for cancer. I have 200,000 tissue in a tissue bank sitting in Tata Memorial Center. I'm just looking at scanning those samples. I don't know what I'm looking at. The pathologist, who is my colleague, Swapnil, doesn't know what he's looking at because new kind of data. I'm looking at UV 3-dimensional data sets that nobody has ever looked at before. But the hope is that we'll be able to make sense out of it if I measure lots of different parameters. Okay, so ultimately, it doesn't negate data collection. It's not a solution that I can just sit and play with my younger son at home and things get done. But I think more data collection. We need a lot more data for things where there is no data set that exists. But then, yes, go back to AI in an intelligent manner and find out what you want to find out. So let me thank the people who have done this work. Everybody from Parijat onwards to the Anup and Ankur who are yet to graduate, but I'll just mention the few who have mentioned in this work. Anirban and Anup did the folding modifier work. Arpan did the single molecule oligomer study. It was started by Shimli, but what I talked about was that. Serotonin membrane interaction, I didn't talk about Dev Shankar's main work, but there are lots of other people involved in it. Thanks to all my current and past students. What I can't really express is that being in TIFR, nobody breathing down your neck, you can be free. Even the students are wonderful students. You tell them, look, doesn't that look interesting? And I say, yes, Sudipta, that looks interesting. And we go ahead and do it. Then don't question, oh, but this, why that? You think of that. The last part I didn't talk about, and literally the whole field opened up. It's still a small field, but to us, a few others are doing it now, standing in front of the board in D304 out there. So this is really wonderful because you have such wonderful students and postdocs and scientific officers. Also, you get a lot of good collaborators. I thank Mahalakshmi, Aisar Bhupal, Michael Schlier, TU Dresden, Devashish from here, Amitesh from here for the bacterial work, Anna and Shachi Gosavi, Kollaneshwar Mandal, and Ranibari Das from Shachi and Ranabir from NCBS, and Kollaneshwar from TIFR Hyderabad. Madhu, Daniel, and Ravi are my collaborators of this little part. I don't know, I have a list of 30 collaborators who have contributed different parts. And membrane interaction with Daniel, Vidita, Maisuan Lee, and Pavel Krupa from Poland. And funding comes from both TIFR through DAE through TIFR, DBT, Ministry of Information Technology, and the Wellcome Trust. And thank you very much. Sorry, I ran out of time.
Thank you, Sudipta, for this wonderful talk. Colloquium. I'll go with the student first.
I just had one question about that fact that you designed something which was kind of preventing the complete folding from the intermediate. So in case of antibiotics, the structure of the protein changes to kind of prevent the action.
Right.
Do you think that the folding speed itself can be evolved to become, because superfolder was something that evolved from GLP.
From there, yeah.
So can proteins evolve to fold faster to avoid that problem?
That was our first target. And we met the target with the first paper. It was just looking at folding. If the thing goes in, it's a nucleus. So to form the folding nucleus takes time. If you preform the nucleus and it grabs around it, the folding becomes faster. That's all we showed in the first paper. Okay. And the only paper that has been published till now. The two which are following, we're looking at the function. Yes, it folds faster.
So AlphaFold, it does not do a good job in predicting intrinsically disordered regions. So I was wondering, is there any way to predict IDRs or a so-called formula which can help us predicting IDR structure?
Okay, so if something is almost fully disordered, like amyloid beta monomer, there's a meaning to the word structure. I mean, what are you going to predict? Okay. However, most of these proteins actually are partially structured, or occasionally, maybe 30% of the time, they're populating some kind of specific conformation, other 70% they're not. So there are now prediction things, not using real AI, but preceding that AI, there are some algorithms by which, again, looking at local structure, local interactions, putting that information in, trying to predict that. They frequently get it wrong. And still now, there is no AI program that gets it right. But as I said, many of them are so unstructured, there is no meaning to the word structure.
So the Q-slip data, what is it telling? Is it giving the distance of the quencher from the fluorophore? I mean, how deep is your peptide going into the membrane? Or
right. So let me just draw it. So I didn't do a good job as in a hurry. So what you're doing is you're asking that you have protein, okay? And you put a reporter somewhere, let's say at the tail here. And you think that it is going into the membrane, which is 4 nanometers thick, okay? Okay, 4 nanometers. And you want to ask this part, how deep in the membrane does it go? What you have done is you have put a little quencher right here. So if this guy, and it needs to collide to quench, not a FET quencher. So if this guy at least a substantial percent of the time is in touch with this layer, so it actually is not here, the way I have drawn it, it will never quench. But if I put the quencher in this level, so there are, of course, all the lipids have this quencher, or at least say much of the lipids, and it's at that level, and the lipids go in. I put it here. I know exactly where I put it. It's synthesized like that. So if the quencher is close to the fluorophore at the same depth, this depth, I know that that part of the protein is going to that depth. If I put the quencher here in a different preparation, then it doesn't quench. I know it's not going.
But will the quencher, like you have two things here now artificially introduced into one is your peptide, which is supposed to be toxic, has a fluorophore, and your lipid also has a quencher. Would it not affect the properties? And then in that case, wouldn't something like MD, which could be done in a membrane without the fluorophore or the quencher, give you better information?
Very good question. In fact, that was the first thought with which we went for checking most toxic, partly toxic, and non-toxic. And we see that the most toxic, A-beta 42, these are valid points. They may behave differently. The most toxic, they have very different characteristics of entry. And though their differences are single or maybe 2 residues. And B, that the more toxic it is, the deeper it enters. So I already know the toxicity, but it enters deeper. So there is a correlation with by checking with toxicity, which says that perhaps it's not destroying all the quality.
So your higher Q-slip values means it's moving deeper. So the red ones were higher Q-slip is
red ones were
on the
A-beta 42, the highest toxicity.
And that's
green
on
ones
the top.
were A-beta cyclohexyl-aniline, the lowest toxicity.
Oh, so the distance is basically with y-axis increasing? Or
Y-axis, so there was C16, which is at the middle, as you know. So A-beta 42 enters the deepest, so the C16 gives a very large Q-slip. Right. Right. So depends on how fast the protein folds, because an external peptide will have to collide with it. So it depends on the concentration and diffusion constant. Small peptide will have some standard diffusion constant. So you want it to collide with it before it has folded. So that's why you're talking about this targeting the parts that fold the last. In a beta barrel protein, which is especially folding in membrane, this part, which is I call the seam, would be folding the last. So I'm trying to buy time for my peptide to go and attack. But yes, if it's a very fast folder, then it won't be very effective.
Yeah, hi. So my question is more of a confusion. So you said that you have an unfolded protein and then you put in some environment and you say that this is the interaction and the computer folds itself, right? Yeah. So then my question is that if the computer is going by, this is the interaction and then, so how do we not know the dynamics? So that
now, when you do the MD simulation, you do know the dynamics.
Okay.
I'm talking about AI not knowing the dynamics.
Okay. So how does the AI then does? Yeah.
Oh, so it's similarity.
Okay.
So that's one word, sort of similarity. It has a training database of, it's much more sophisticated than that, as we found out through this half a day long interaction with John Jumper. It's much more sophisticated than that. It does some local MD simulations, etc. But basically, the first thing is similarity. If it has seen 100,000 structures, at least by parts, and one is little alpha helical, it has a probability, etc. It first goes there. Then it does local MD simulation. Then it.
Thank you.
But similarities.
And there's one question. When you showed this red triangle, which is interfering with the holding of the green triangle.
Right.
In the wedge.
Right.
So that's ultimately a small molecule kind of thing. Okay? Or can I extrapolate this kind of concept to interfering with amyloid sheet formation?
That's what we like to do. It's not a small molecule. It's actually a part of the protein itself. It's about, depends, 20, 25 residues long. So it's definitely not a small molecule. It's a medium-sized peptide. So we look at the barrel, for example. Take the seed. You already know the seed. The structure you know, the sequence you know, and there is the other part of the seed. You just join it a little bit here, join it on the top, make a loop structure, and put it in. And you check that it is folding into, let's say, an anti-parallel beta sheet kind of thing, and you put it in. So that's 28 residues, I think. So it's not a small molecule. That's the beauty of it. Is also the sensing is distributed, senses everywhere. So it's not easy for the bacteria to mutate one place and do something.
Okay. Okay. Sorry about that, Sudipta. But I think we will end the colloquium here, but this is fantastic. Sudipta, thank you for coming back and presenting us so nicely all the work that you and your students have been pioneering in. And I am so glad that some of the oldest members of the group are here. The family was here. And Alak was always he comes back and asks questions. He's always there in colloquium. So Alak, thank you for coming back.
And let me thank my TIFR colleagues and friends. You'll always remain my colleagues and friends. I invite you to Hyderabad. Do visit BITS. Also the biophysicists, as the president of the Indian Biophysics